In this video, we're going to talk about tonicity and osmoregulation. We're going to learn about water potential, solute potential, and maintaining water balance. So to start off, who led the Israelites through the semi-permeable membrane? That's osmosis. Osmosis is the diffusion of water through membranes. Most water moves passively through a membrane, but we can have large quantities of water move through a membrane through proteins called aquaporins. When we think about water moving through a membrane, the cell has to maintain dynamic equilibrium. And that means there are equal amounts of water moving into and out of the cell at any point in time. Water will move back and forth equally between the two sides of a membrane, so that there is no net movement of water between the two areas. That means that water doesn't stay on one side of a membrane or the other for an extended period of time. It simply moves from one side back to the other and back again, back and forth. And water will flow across a membrane until it reaches this state of dynamic equilibrium. So imagine we have a membrane, and on the two sides of the membrane, there's a gradient of water. What will happen to that water? It will slowly move down its water concentration gradient from a high water concentration to a low water concentration until the concentration of water on both sides of that membrane are equal. That means the cell has reached dynamic equilibrium. Cells and other body tissues always have a certain amount of solutes and a certain amount of water. And we can measure that amount by using the term molarity, which is just the amount of solute that's dissolved in some solution. So if we take a solute and we dissolve it in a solvent, what we get is a solution. For example, if we take some dye, put it in some water, and let it dissolve, we get a purple solution. One example that I'd like for you to know about is distilled water, which sometimes we refer to that as pure water. And that is different than tap water. Tap water has solutes, like ions and minerals, dissolved in it, whereas pure water or distilled water does not have any solutes dissolved in it. Tap water has about 500 parts per million of solute versus distilled water, which has 0 parts per million. And when we want to describe the relative concentrations of solute inside or outside a cell, we use the term tonicity. That term tonicity can be used in 3 ways. We can say that a solution is hypotonic, isotonic, or hypertonic. Solutions that are hypotonic typically have a low solute concentration. Solutions that are isotonic have an equal solute concentration. And then hypertonic solutions have a higher solute concentration. Those 3 words only make sense when they're used to compare 2 environments. That could be the inside of the cell compared to the outside of the cell, or vice versa. For example, here we see that the environment is hypotonic to the cell. That means that there is a lower solute concentration and higher water concentration outside the cell than inside the cell. Whereas in this circumstance, the environment is isotonic to the cell, and that is because there are equal concentrations of solute and water on both sides of the cell. And then lastly, we have the environment is hypertonic to the cell, and that means that there is a higher solute concentration and lower water concentration outside the cell compared to inside the cell. So in these 3 circumstances, what I want you to see is that we use the term isotonic, hypotonic, and hypertonic to describe 2 environments, or really the relationship between 2 environments. And we have to always state which 2 areas are being compared in order for those terms to make sense. Here's a practice question. Is the environment hypertonic, isotonic, or hypotonic to the cell? Take a second to think about your answer. And I hope you said that the environment is hypotonic to the cell. One thing you could possibly be asking yourself is, could we also say, is the cell hypertonic, isotonic, or hypotonic to its environment? And the answer is absolutely. You can ask that question. The answer would be that the cell is hypertonic to its environment, and that's because there is a higher solute concentration inside the cell compared to outside the cell. Take a second to look at this checkpoint question and see if you can come up with an answer. I hope you said that the salmon is moving from a hypertonic environment to a a hypotonic environment. And that's because the marine salmon lives in the ocean, which is a hypertonic solution because it has salt in it, versus a freshwater stream is a hypotonic environment, and that is because there is a lower concentration of salt compared to the ocean. Don't forget that these terms only make sense if you explicitly state both areas that you're comparing. Whenever water moves, it wants to move in the direction that it can help dilute areas that have a higher solute concentration. So, for example, water will move from a low solute concentration area to an area with a higher solute concentration. That means that it's moving from a hypotonic solution to a hypertonic solution. You can remember this because hypo, water must go. Water will go from a hypotonic environment to a hypertonic environment. Take a look at this figure. Here we see that a plant cell, which has solutes contained within, is being placed in distilled water. Remember, distilled water contains no solutes. So, distilled water will be a hypotonic solution compared to the stuff inside the cell. If we place that plant cell into the distilled water, can you predict where water will go? Will it exit the cell and go into the test tube, or will water from the test tube enter the cell? Remember, hypo, water must go. So where is it going to go? The water will enter the plant cell to help dilute the higher solute concentration inside the plant cell. Over time, osmosis will stop. The water will enter and leave the plant cell because it's at a state of equilibrium. A question I have for you is there a limit to how much water a cell can hold? And to answer that question, I want you to think about the last time you saw a snail or a slug. In this case, we have a snail. What happens if you take some salt and put that on the snail? RIP. The snail will become so dehydrated as water leaves the cells that make up that snail and goes out of the snail to help dilute the salt concentration. Imagine we have a bag of red blood cells, and if we take those red blood cells and place them in different concentrations of solutions, they will either burst open or we'll see that they shrivel up depending on how much water enters or leaves the cells. For example, if we take one of those animal cells and we place them in a hypotonic solution or a very hypotonic solution, such as pure water, the cell will take up water by osmosis because the water wants to move to an area where it can dilute solutes. And if there are solutes floating inside those red blood cells, water will want to move from the very hypotonic solution or the hypotonic solution into those cells. And as water enters the cell, more and more pressure will build up until the cell becomes swollen, or even until it lyses, which means the membrane bursts open. Interestingly, for plant cells, they prefer this kind of hypotonic environment since that means water pressure will build up inside the plant cells and keep the cell membrane pushed up against the cell wall. And we'll talk about this later. In isotonic solutions, water will enter and exit the cell at similar rates. And this is what is preferred by animal cells. And then lastly, if we have hypertonic solutions, water will exit the cells, which will cause the membrane to lose its internal pressure, and then the cell will become dehydrated and shrink in size. If you've ever had to get intravenous fluids, you may have wondered why they use sodium chloride in the IV bag. And the reason is because 0.9 sodium chloride is isotonic to human cells. It won't cause the cells to swell or shrink, and instead, the water will enter and exit the cells at similar rates so that those cells have just the right shape and volume. This sodium chloride solution is also called saline, and it's designed to match the tonicity of our body cells, ensuring that all of our cells remain properly hydrated. If you think about plants have internal structures called vacuoles, which are known to store water and solutes. If the vacuoles are filled, then the filled vacuoles will put pressure on the cell membrane and the cell wall, and that will keep the plant turgid, or plump and firm and stay upright. And if the amount of water inside the vacuole goes down, then plants will lose their turgor pressure and the leaves will become wilted and the plant will eventually die. So when we look at a plant and see it has high turgor pressure, we say that the plant has turgid or is turgid, versus a plant that has a low turgor pressure, it becomes flaccid. One way we can think about the movement of water is that water moves from a high water concentration to an area of low water concentration. Those terms represent the same relationship that we have when we think about concentration gradients with solutes, except in this case, we're talking about water instead of solutes. We can describe this relationship between high water concentration and low water concentration with a different set of terms. We use the term water potential when we're thinking about the concentration of water. And water will always move from an area of high water potential to an area that has low water potential. In terms of solute potential, we have to think about solutes and the way they interact with water. Solutes have this pulling effect on water, and the direction of water will always be from an area of low solute concentration to an area of high solute concentration. So water will move by osmosis into the area that has the higher solute concentration in order to help dilute the concentration of solute on that side. When we write solute potential, we use the symbol psi sub s to represent that. And a solute potential just represents the pulling effect of solutes on water. It's what directs the movement of water across the membrane. And the direction of water movement will always be from an area of low solute potential with fewer solutes to an area of high solute potential with more solutes. And that's a bit counterintuitive, but remember, water is moving towards the area where the solute concentration is higher. And an interesting aspect of solute potential is that the value is usually negative. And this is because solutes reduce water's ability to move. A water molecule would need to somehow overcome and then resist a solute's pulling effect in order for that water to get separated from the solute. Let's watch a video that represents this phenomenon.
Osmosis is a very well-known phenomenon with a less well-known explanation. Osmotic flow occurs when two volumes of water are separated by a membrane that allows the passage of water molecules only, but not other molecules. If the water on one side of the membrane has a high concentration of dissolved particles, called solute, water will flow in both directions across the membrane, but more will flow in the direction where the concentration of solute is higher. This process can create enough pressure to burst a cell, and routinely creates the pressure that holds plants upright by creating cell turgor against the sturdy cell walls of plant cells. The movement of water to a region of higher solute concentration describes what happens, but why does it happen? On a molecular level, the solute has a special influence on the water very close to the barrier. Water molecules and solute exist in a world of constant molecular collisions. At the barrier, water can pass through in both directions, but the solute molecules collide with the barrier and bounce off. The rebounding solute collides with water molecules, transmitting kinetic energy and driving water molecules away from the barrier. It is as if the barrier has a repellent force field on one side only, pushing water molecules away. Multiply this effect by billions of solute molecules in constant collisions with the barrier, and the net result is that more water flows in than out, and osmotic pressure builds.
A question I have for you is, what is the water potential of pure water? In this case, distilled water has no solutes. And since there are no solutes, that means that there is no solute potential. In other words, the solute potential for pure water will be zero. And if we assume this is an open container, the pressure potential for this will be zero. So that means that the water potential will be equal to zero. What we're looking at is the equation for the solute potential of a solution, which is provided on the formula sheet for the AP exam. And the equation for solute potential is psi s equals negative I CRT. Each of those values represents a different important component of calculating the solute potential. So for example, I stands for the ionization constant, which will always be equal to 1 if we're using sucrose, or it could be equal to 2 if we're using a salt like sodium chloride. The letter C represents molar concentration. The letter R is a pressure constant, and it will always be 0.0831 liters times bars per moles times Kelvin. And lastly, the T stands for temperature in Kelvin. And the way we calculate Kelvin is just by taking whatever the temperature is in Celsius and add 273. One thing you want to notice is that if we add more solutes to a sample, that will cause the solute potential to become more negative. Here's an example I want you to try on your own, and then we can review. So for the first question, which side has a lower solute potential? I hope you said that the solute potential of side A will be lower or more negative than side B, and that is because solute potential becomes more negative as solute concentration increases. For number 2, where will water move? I hope you said that water will move from side B to side A, and that's because water will move from area of high water potential to an area of low water potential. In other words, water will move to help dilute solutes. And the last question, the water potential of side A is -3 and side B is 0. How will increasing the water potential of side A by 3 bars affect the net movement of water? And so the answer to that is there will be no net movement of water between the two sides, as the two sides will be isotonic to each other. Because if side A was originally -3 bars and we increase it by 3 bars, that will mean its new water potential is 0. Let's explore how water potential plays a role in plants. One of the most striking features inside plant cells is the vacuole. A vacuole isn't just a storage unit, it plays an important role in maintaining the plant's turgor pressure. How does it do this? Plants have a nifty strategy of pumping solutes into this vacuole. And as the number of solutes increase inside the vacuole, the water potential inside that vacuole becomes more negative. Let's imagine the water around the cell. With our plant cell vacuole labeled at -10 bars and the surrounding environment at -5 bars, it's clear that the water potential inside the vacuole is lower than its surroundings. This difference means that water will naturally flow into the vacuole, trying to even things out. As the vacuole fills with water, it expands, and as it does, the cytosol and the plasma membrane are pushed outward, exerting pressure on the cell wall. This pressure gives plants their rigidity and is crucial for their upright growth. If you want crunchy celery, try putting it into distilled water. Can you think of why that would make it more crunchy? Meet paramecium. Paramecium is a single-celled organism that lives in freshwater environments, such as Blackfriar Pond. The interior of a paramecium is hypertonic to its environment, meaning it has a higher concentration of solutes. This difference in concentration causes water to flow into the paramecium via osmosis. But what prevents this paramecium from swelling up too much? The answer is its contractile vacuoles. These structures temporarily store excess water and expel it, preventing the cell from bursting or undergoing lysis. Now, consider a scenario where paramecium finds itself in a saltwater environment. With a higher concentration of solutes outside, the osmotic balance shifts. What do you think would happen to the paramecium in this hypertonic environment? I'll let you ponder this question. For organisms that live in a hypotonic environment, such as freshwater fish, they need a way to prevent their cells from taking in too much water by osmosis, similar to a thirsty sponge. So these kinds of freshwater fish will regulate the amount of water going in and out of their bodies by drinking less water and by producing very dilute urine with low concentrations of salt and high concentrations of water. On the other hand, hypertonic environments such as the ocean will constantly try to dehydrate and pull water from the saltwater fish. So for fish that live in the ocean, they need ways of removing excess salt by releasing it from their gills and by producing very concentrated urine with high levels of salt and low levels of water. These processes are both examples of osmoregulation, which is the way we maintain proper water balance and salt concentrations. Take a look at this question and see if you can select all of the correct answers. And then try this multiple choice question. Finally, what do you think of this question?